Today we are going to talk about neuromuscular junction, the definition of neuromuscular junction, its anatomy, and the mechanism through which impulses are conducted from the axon to the musculature through it. So first of all, what is neuromuscular junction? It is a junction through which the nerve impulse from the axon is transmitted to the musculature, leading to release of calcium ions and in turn leading to the muscle contraction. As far as its anatomy goes, it has a trough-like area in between it. This is known as a synaptic gutter or trough. While there are grooves and hills at the borders, these are known as subneural clefts. Now, what actually is the benefit or the function of subneural clefts? It increases the surface area through which the neurotransmitters can act. So, for example, if the muscle have large subneural cleft, it means that the impulse will be transmitted more strongly and the action of impulse will be higher than that of any neuromuscular junction having low subneural clefts. Now, if we zoom in into this junction, it shows that the subneural clefts have receptors and Na-gated cerium or sodium-gated channels. Now, the receptors are acetylcholine receptors, meaning they are sensitive to acetylcholine. The acetylcholine receptors are different in infants and adults. For example, in infants, the receptor consists of 5 proteins, 2 alpha, 1 beta, 1 delta, and 1 gamma, while in adults, the gamma protein is replaced by epsilon. Now, how do these receptors work? Well, actually they are constricted. They do not let the influx of sodium ions inside. But when the impulse is transmitted, the 2 acetylcholine receptors attached to 1 alpha protein each, leading to the opening of the acetylcholine receptors and in turn leading to the influx of sodium ions inside the subneural cleft. Now there are a few points which will help us in the upcoming topics. These are acetylcholine is excitatory neurotransmitter. It is not an inhibitory neurotransmitter. The second point is the action potential of a skeletal muscle is slower and long lasting than that of the neurofibers. This is due to the functional difference between the axons, the neurons, and the muscles. The muscles require stronger force, stronger impulse, but they also need long-lasting impulse. And the third point is the resting membrane potential of skeletal muscle is minus 80 to minus 90 millivolt, which is more negative than that of the neuron, which is minus 70 millivolt. Now the way through which the impulse is transmitted from the nerve fiber to the musculature through this junction is really simple. So first of all, the nerve impulse is transmitted from the brain, from the spinal cord, wherever it goes. Then it goes to the axon terminal, and in axon terminal it opens calcium-gated channels, which will lead to the influx of calcium ions into the axon. This calcium influx will cause the activation of calcium calmodulin dependent protein kinase, which will in turn phosphorylate synapsin proteins, which binds these vesicles here in its place. Once the vesicles are free to move, they will move towards the boundary of the vesicle or the membrane of the vesicles. Now the vesicle presents here, which are bounded by synapsin proteins, these are called synaptic vesicles. They consist of a bunch of 100 type of neurotransmitters. Once the synaptin proteins are phosphorylated, the vesicles are free to go. Now they float towards the boundary of the axon where they release their content, which are, for example, in this case, acetylcholine. Once the acetylcholine are released, they will flow to the subneural cleft where they will be attached to the acetylcholine receptors. Now, once the acetylcholine receptors are attached to the subneural cleft's acetylcholine receptors, the conformational changes will occur in these receptors. They will allow the flow of positive ions and will block the flow of negative ions. This is due to the amino acids in the acetylcholine receptors which are highly negative. Thus, it will block any negative ions to enter the subneural cleft and in other ways allows sodium ions or positive ions to enter. Now, when the positive ions enter the subneural cleft, they will cause a rise in potential. This rise in potential is known as end plate potential. Now, due to high end plate potential, more and more sodium enters the muscle cells through the sodium gated channels, including those present in transverse tubule. Now, what are transverse tubule? We will find out. The action potential develops at surface of muscle, but for it to penetrate deep, transverse tubules is required, which are actually internal extensions of cell membrane. So if cell membrane at subneural cleft is depolarized, then the T-tubule membrane is also. Now this here is a muscle bundle. As you can see, these are muscle fibers and the yellow projections here are cisternae of the cytoplasmic reticulum. The tube here that connects all these muscles and the outside environment is known as the transverse T-tubule. The function of transverse T-tubule is the flow of sodium channels into the muscle membrane deeply enough to excite all the muscle fibers. Now, once the action potential has risen enough to the end plate potential, sodium influx will occur, which will causes even more excitation of the membranes. In turn, this will cause the DHP receptors to excite. Now, when the action potential has risen enough to the end plate potential, the dihydropyridine receptors will excite or they will be activated, which in turn will cause the opening of calcium plus 2 release channels, or we can say as ryanodine receptors. Upon opening, efflux of calcium plus 2 will occur and they will enter all into the cytoplasm of the muscle. Now, once the muscle contraction has run its course and repolarization is about to occur, during repolarization, the calcium in the muscle cytoplasm will be retracted back into the sarcoplasmic reticulum. Now, how does it occur? What is the mechanism of it? Well, actually, the sarcoplasmic reticulum has a pump which is known as sarcoplasmic reticulum ATPase, or we like to call it SIRCA. Now, the calcium will be uptaken through the SIRCA into the sarcoplasmic reticulum. Once there, they will bind to a protein called calciquestrin, which will bind the calcium ions inside the sarcoplasmic reticulum and enabling its release. Now, what actually happens to the acetylcholine when the action potential has subsided? So, to know that, first we have to know about the synthesis of acetylcholine and then we can talk about its destruction. Now, first of all, the acetylcholine vesicles are formed in the cell body of the neuron, which is then transported through the axon to the axon terminal. Here in mitochondria, acetylcholine are formed and they are packaged into the vesicles. Once in the vesicles, when the action potential arises, they will fuse to the membrane and will release its acetylcholine receptors, which will perform their function just like I have explained before. Now, once its function is over, the acetylcholine esterase, which are actually enzymes, will cause the cleavage of these neurotransmitters and they will be converted to acetate ion and choline. Now the choline will be packaged back into the terminal, which will be used to form the acetylcholine receptors back and back again. While the vesicles will be formed from the cell body as well as from the detachment and inward movement of this membrane into the axon terminal. That's all for this video, folks. If you like it, Please do subscribe, share, hit the bell icon for further notification, and let me know which topics you have confusion it, and I will cover it in the upcoming videos. Thank you very much.